Hello everyone, my name is Uthayavignesh. Today I am going to present about my industrial training presentation at United Plantation Berhad or UPRD, United Plantation Research and Development. And my mentee number is f22b0159 and my supervisor's name for internship is Dr. Kanda Sridharan. The most valuable experience during my internship is I gained hands-on experience by collecting the morphological data of coconuts from 12 coconut varieties using Cogent Stantec descriptors as a guideline. And this research also deepened my understanding of plant science and germplasm research. And I also conducted fruit component analysis or called FCA on the coconuts in a professional laboratory setting at UPRD. And this has strengthened my laboratory skills, precision in measurement, and self-discipline and scientific discipline. Speaking about the most memorable memory during my internship is this picture. These are the people who helped me to do the whole project. For the acknowledgement, I would like to give my sincere gratitude to my supervisor, Dr. Kanda Sridharan, the research manager, breeding department of UPRD, for his invaluable guidance and support and mentorship throughout the industrial training and this journey. And also my heartfelt appreciation to Mr. Saran Rao, son of Lechmaya, assistant research officer at UPRD, also guiding me and assisting me throughout this project until the statistical analysis. Let me give you a brief introduction about United Plantation Berhad. So, United Plantation is a Malaysian Danish plantation company founded in 1906, centered at Jenrata Estate, Bagandatu. And it operates vertically integrated business model from seed to market. And they have engaged in plantation management, processing, and research and development (R&D). And they also globally recognized as one of the certified sustainable palm oil producer, and also was the first company ever to certified by the Roundtable of Sustainable Palm Oil (RSPO). And United Plantation also one of the largest collectors and conservators of coconut germplasm in Malaysia after FRIM and Agriculture Department of Malaysia, and maintaining a diverse coconut genetic resource collection for breeding and their research purposes. So their main focus crops are oil palm cultivation of 90% and 10% of coconuts. So these are the group structure for United Plantation. For plantation, we have United Plantation, and at Indonesia, we have PT Surya Sawit Sejati. And for palm oil refining, we have Unitata and Unifuji, and others will be under Burnham Advisory Services. So speaking about UPRD, United Plantation Research and Development background, it was started in the 1950s in the collaboration with Department of Agriculture of Malaya, and UPRD was officially established then in 1962 at Chandrata Estate, and because to centralize and strengthen the agriculture activities, research activities. And over the years, UPRD has developed into a modern research centers with advanced facilities like molecular laboratories, tissue culture labs, soil and leaf analysis labs, and bunch analysis labs. And these efforts have resulted in United Plantation being recognized as one of the highest yielding plantation companies in the world. So the key achievements of UPRD is they have developed high yielding oil palm planting materials (DXP), which is Tinera, and contribution to making UP one of the highest yielding plantation companies in the world, as I mentioned before. And they also have developed successful crop improvement programs for oil palm, coconuts, and bananas, and also their continuous innovation in plant breeding and crop production and sustainable agriculture. So it's been 100 years they have involved in the research and development. So these are the activities at UPRD. They do research and services. For the research, they do crop improvement, crop production, crop protection, and plant propagation and byproduct utilization, and also environmental issues and crop sustainability. For services, they do oil palm seed and seedlings, as I mentioned before, and then coconut seed nuts and banana ramex, and also ornamental landscaping and forestries, and also super growth of grow and bajaria and advisory services and feasibility studies. This is the organization structure for UPRD. So for the Director of Research, Mr. Ho Xiu Hing, and Research Controller, we have Dr. Vijendran. And for Research Manager, we have research manager for breeding, we have Dr. Kanda. And research manager for tissue culture, we have Mr. Wo Fu Hin. And senior assistant research manager is Ms. Madam Dalila Binti Dawood. And senior research officer is Mr. K. Kamlesh. And there are four research assistant officers, who is Mr. Saran Rao, Ms. Nur Laili, and Mr. Choi, and Ms. Anis Ashikin. So before I getting started to talk about my project, I would like to give a small introduction about coconut and its morphology. So coconut or Cocos nucifera is from Aracaki family, same as the oil palm, and it has diploid chromosome numbers, which is easy for crossing. They shares half of the mother's chromosome, father's chromosome, and coconut also monoecious plant, which means they have male and female organs in their same inflorescence. For my morphological analysis, I have collected the characteristic of stem, leaf, inflorescence, and then fruit component analysis, and then the fruit appearance for the coconut. So I will let you know in the further slides. So coconut has a fibrous root system, and speaking about the stem, they are single unbranched woody stem, as you can see in this picture. And for the leaves, they have people call it feather-like leaf compounds, and you can see in the picture here. So for the inflorescence, as I mentioned before, coconut is a monoecious plant. So they will have a male flower and female flower in their same inflorescence. So these are the parts of the inflorescence. We have spadix, female flower, first rachilla, peduncle, male flower, last rachilla, and the spathe. So speaking about the coconut fruit, the outer layer is called the exocarp. The middle layer, which has the husk part, is called the mesocarp, and the hard shell layer is called endocarp, and the coconut meat is called endosperm. And the water, of course, we call it coconut water. And then we can see embryo or germ in the coconut fruit. So this is the morphology of coconut fruit. So as I mentioned before, I have collected coconut classifications, which is six talls and six dwarfs. So there are characteristics for each varieties for tall and dwarfs. So for the tall, they are normally cross-pollinated. As I mentioned before, the male flowers will open early comparing than the female flowers, and this will encourage them for cross-pollinating. And because they are cross-pollinating, they have a high genetic variability and longer lifespan. So taller coconuts can live along 80 to 100 years if the environment is suitable. Speaking about the dwarfs are mostly self-pollinated, and it is because the female and male pollens will open at the same time. And because they are self-pollinated, they are more uniform, and we can notice the early flowering in the dwarfs. So why this project, morphological analysis of genetic diversity in coconut, is important is because it supports the germplasm conservation, and for the breeding programs, they have kept the details and the data and will help readers to refer this data. And all the morphological traits are based on Cogen Stantec descriptors. So the objective of the project is to collect the morphometric data from different coconut germplasms and to characterize coconut germplasm using Cogen Stantec descriptors. Speaking about the Cogen was established under the International Plant Genetic Resources IPGRI in 1992 and consists of 39 coconut producing members of the country. It represents more than 98% of the global coconut production. And Cogen also supports international collaboration in coconut research and breeding in the future. And they promote the conservation and utilization of coconut genetic resources. Cogen also protects the coconut biodiversity and climate change, pest and disease, and urbanization. So for the methodology, for the leaf methodology measurements, I have these methods, which is for the petiole color, I have to visually rate them according to 1 to 11. So you can see the color rating there in the table. And for the petiole length, I have to measure them from the base to the most proximal leaflet. And for the petiole white, same as the petiole length. And for the petiole thickness, I have to measure at the first insertion of the leaf as per the picture. So for the rachis length, I have to measure them from the base of the petiole to the leaf tip. And I also have to count the number of leaves for each palm, each leaves. And I should count them on the right side of the leaf. So I also have to measure the leaflet length and leaflet width. So for the leaflet length, I have to pick the middle leaves left and right on the rachis. And then I have to measure them like this. And for the width, I have to measure them like this, and then I have to take the average and note them for the SAS analysis. So for the girth or the stem measurement, I have to measure the girth at 20 cm above the soil level to check the base size of the specific variety. And for the length of the 11 leaf scars or ring-like structure, I have to count them 1 to 11 and measure the length, and this will help me to define the age of the palm and the leaf production of the palm. And for the girth at 1.5 meter, I also have to measure them, and this is to standardize the middle part by avoiding the soil and other disturbance. So for the inflorescence, I have to measure the length of peduncle, length of the central axis, diameter of the peduncle, number of spikelet with and without female flowers, length of the first spikelet bearing female flower, total number of spikelet, number of female flowers, and female flower distribution per spikelet. So every samples, every parameters, we have to do 30 pumps per variety. So these are the examples of the methodology for inflorescence. You can see the length of central axis, the length of peduncle, and here you can see the male flowers are open. So for the fruit appearance, we have equatorial and polar view. For equatorial, we have 1 to 5 rating, and for polar view, we also have a 1 to 4 rating. So I have to rate them according to these shapes. So for the fruit set, I have to visually estimate the number of fruits bigger than feast in each samples of the palm. And for the fruit color, same as the petiole color rating. Here you can see the table. And for the, I also have to see the shape of the dehusked nut. We have the rating 1 to 4. So this is the example of the dehusked nut. So for the fruit component analysis, I have to check the fresh weight, nut weight, weight of split nut, shell weight and meat weight, which is a difference between shell weight and the meat weight of the split nut, and also measure the endosperm thickness, which is measured at the equatorial portion of 10 mature fruits, and the mean value will be calculated. So these are the examples of the FCA. Also, I have done this 30 coconuts or 30 samples for each varieties. So speaking about the tall and dwarf varieties, there are hybrids also. Hybrids like Matak, which is crossing between the varieties called MYD and MRD as their mother palm, and Taknanan tall, which is known for its water capability, water saving capability, and fruit bunches capability. Also, we have Mawa, which is created by UP itself, and also Matak also created by UP in a collaboration with the Jabatan Pertanian. So for the Matak, same, the mother pump is MRD and MYD, along with West African tall, which is known for its coconut meat thickness, and we can get much more better coconut milk from the Mawa variety. So what are the tall varieties I have collected? As I mentioned before, Tatanan tall, West African tall, Renal tall, Laguna tall, and then Tahitian tall, and also Malayan tall. So for the dwarfs, I have taken the MRD Malayan red dwarf, MGD Malayan green dwarf, MYD Malayan yellow dwarf, Pandan green aromatic dwarf, Takuna dwarf, and Katigan dwarf. So let's see what are the difference between tall and dwarf varieties. So for the growth habits, tall varieties have a larger leaf and longer edges and more overall vigorous growth. Meanwhile, for the dwarf, we have a small and more compact growth. For the flowering type, predominantly male flowering we can see in the tall varieties, and that is why mostly are cross-pollinating. Meanwhile, for the dwarf, overlay, which means a male and female pollens open at the same time. And for the trait, which is a coefficient variance, which I have done the SAS analysis, uh, tall varieties shows a higher variability across most traits, and dwarf shows a lower variability and more uniform across all the palms. And this is why dwarf are preferred as a mother palm because of their uniformity. Speaking of the fruit yield, tall varieties have a generally heavier fruit, nut, and a husk and shell weight. Meanwhile, for dwarf, it always smaller than tall. For the trunk, tall has a larger as overall, and for the dwarf, we have very smaller. For the inflorescent, the tall varieties has more developed spikelet counts in some varieties. Meanwhile, for the dwarf, generally have smaller inflorescent, but they are known for its early flowering behavior. So, as a conclusion, this project helped the germplasm characterization serve as a foundation for any effective coconut breeding and conservation program in the future. Besides, they had needed an intern for this project to kick off, and then they will continue in their future programs. It will help them for their breeding programs in their future. And also, this study provides a preliminary but a valuable step toward the understanding and supported by the future field trials. Hybrid evaluation and molecular analysis is also needed for the future studies since it's preliminary. So my suggestion for the faculty should incorporate more practical and field-based learning components into the curriculum before the student comments into the industrial training. It will make the students to more easier to expose to the working environment and to the field. And also can prolong the internship period like we have 3 to 4 months, can longer to 6 months to 8 months because we can learn more. So suggestion for the industry is industry should organize exposure visit to other departments like tissue culture lab, oil palm processing plant, and molecular laboratory. And industry partners also consider increasing trainee involvement in data analysis, report writing, and research discussion beyond just fieldwork and laboratory testing. So, suggestion for myself is I need to gain more fieldwork experience in coconut and oil palm plantation management to better understand the variety performance under the actual growing conditions. And I also need to gain more exposure in biochemistry, particularly in the areas related to the molecular analysis in the plant biochemistry. So that's all for me. And thank you very much for listening.